We're going to move on to our first keynote. Now, if any of you were awake yesterday, you felt the storms. And so Dr. Rebecca Klaper from the University of Wisconsin-Milwaukee is our first keynote speaker and was hoping to be here today, but flying through O'Hare is not ideal in tornadoes. So instead, she's graciously agreed to join us virtually, so we will have her here in just a second. So Dr. Klaper is the dean and a professor at the School of Freshwater Sciences at the University of Wisconsin-Milwaukee. She studies the potential impact of emerging contaminants like nanoparticles and pharmaceuticals on aquatic life and how we can design these chemicals to have the least environmental impact. She uses cell phenotypic profiling as well as genomic technologies to determine how these chemical stressors may impact organisms and how the biochemical response can dictate sensitivity or resistance to stressors. Professor Klaper is a member of the Wisconsin Academy of Science, Arts, and Letters, a fellow of the Royal Society of Chemistry, and has received a Fulbright Scholarship and a Science and Technology Policy Fellowship. Dr. Klaper received her PhD in ecology from the Institute of Ecology at the University of Georgia. And before I turn it over to Dr. Klaper, I'd like to especially thank her for being here because she was one of the keynote speakers at the very first Emerging Contaminants in the Environment Conference, and she graciously agreed to return for the 10th year of the conference. So thank you for being here, and the floor is yours.
Okay, great. And everyone can hear me okay?
Yep, I can hear you great.
Okay, great. And you can see my slides. Great. I so apologize for not being there today. Mother Nature had other ideas. Even driving yesterday was hazardous. It sounds like it was the same down near you, but people were holding on to lampposts here to walk down the street, so it was a little crazy. I don't think I've ever seen anything like that. But I'm so honored to act as a speaker at this conference. As was just mentioned, I was here about 10 years ago, and I made great connections there and ended up with collaboration, which I'll talk about in this talk. And also, University of Illinois is where I got my bachelor's degree in the honors biology program, and it's near and dear to my heart. So always love coming down to University of Illinois. My research area is on emerging contaminants, of course, title of this conference. And it really started at University of Illinois, where I was really interested in chemistry and biology. And through working through the lab of Dr. May Berenbaum, who was in the entomology department, I realized there was this field called chemical ecology. So I could merge these two things. So I studied them in the natural sense for a while, plant chemicals and how they influence insect populations as my PhD. But as I was graduating, there was this great paper done by the U.S. Geological Survey and Dana Kolpin, who actually was another speaker at that conference, who I ended up collaborating with afterwards, that talked about all these pharmaceuticals that were being found all over the United States in waterways. And it really was the first time I think I connected that everything that we use ends up in our water, and our freshwater systems are so important. And so over the last decade, this field has progressed tremendously, and it's really fascinating to see where it goes. Kind of disheartening to see how many different chemicals we don't know about and what exactly is out there. And so my research area has moved from just trying to measure these contaminants and look at some of the basic toxicological effects to looking at new methods for determining exposure and impact of these chemicals in the environment. When I started, what's great in our facility, it was so great to see the collaborations that your facilities have down there. That's fantastic. Ours, we have the Wisconsin DNR as well as some other organizations in our facility. And one of the things that happened when I first started doing my research is the DNR came to me and said, "Hey, we're seeing this weird thing with perch. And we are not really sure what's going on. There's no males being found, or there's very few males, and the perch are declining in numbers. And it was thought, to be from overfishing to begin with. And then you can see, like in the late 1990s, zebra mussels also came in and ate up the bottom of the food chain. And so people were attributing it to a major decline in food resources for these perch. But one of the things that the DNR started noticing towards the end of this, like 2013 to 2019, is that they were seeing weird things with the gonads of the fish. So on the top right, you can see a picture of male perch, and the gonads are nice and big and white and milky. And then what they were finding was on the bottom, where they were really atrophied and just didn't look right. And so they asked me to come out and do some sampling with them. And of course, as soon as I did that, the perch numbers of males went through the floor. And so we had a hard time even finding males. And they were asking, does this have something to do with the estrogens in the environment that we're hearing about? And so this is kind of where I started with my research, trying to figure out, is there something coming out of the wastewater treatment plant that, and I was focused on estrogens, and that might be causing this. But the problem is there are lots of chemicals out there. And so which chemicals could be causing an impact is a big question. When we actually measured the amount of estrogens, straight estrogens from like birth control coming out of the wastewater treatment plant, it was really very tiny. And if we could measure them at all, of course, technology has improved since then. So you could probably measure them down in the 4 nanograms per liter, which actually has an effect on fish, but we weren't really finding much of anything. And so we started to try to think of what other chemicals might be causing some kind of estrogenic effect on these fish. And as I said, the problem is that there are so many chemicals that we just really don't know enough about. There was a study done by Wang et al. In 2020 where they looked at the number of chemicals that were registered that could potentially be in use and out in the environment. And it was 3 times as many as had been previously estimated. And they didn't really, couldn't identify some of the chemicals because a bunch of them are confidential or the descriptions are ambiguous. And so there's really a ton of chemicals. Even the ones we know about, we don't have enough information, but then there's, of course, a whole host of other ones we really don't know about. And of course, we have limited numbers of tests. There are, this is from David Dix at the US EPA, where they were looking at, how many chemicals do we really have data on in our public databases? And then how much data do we have on some of the sublethal impacts or long-term impacts like cancer or genotoxicity or developmental toxicity or reproductive toxicity. And it really, the amount declines to like, 20% or even down to 10% or lower for some of these. Reproductive toxicity at the time is much lower. And so we might be missing, our assessment of what these chemicals are actually doing. And a lot of these tests are in model organisms, some of which you might not see the impact in those right away. And you're not necessarily measuring all of these endpoints, especially for environmental organisms. We very rarely measure some of these things like cancer causing or the genotoxicity is just in select tests. And so the endpoints might not be there and might not be predictive of what these things do. So there's been a push over the last, 10 years to move towards a kind of a systems approach to looking at toxicity and looking at the structure of the chemicals to which pathways they might impact in organisms, and then tying that into some of the larger scale impacts and data that we have in our databases of big scale impacts. And it's really an impressive thing that's being done by the Environmental Protection Agency and the National Institutes of Health, really linking all of these things together in big, huge databases and trying to model these using also in vitro toxicity. So looking at cell-based assays in order to make some of these predictions. So my data kind of falls within this. We're not a huge lab, so we can't do all of these things, but we really rely on some of this modeling that has been done to inform our own research as well. But it's really an impressive set of work. So what I started doing when I first got to my job is trying to figure out, is there anything impacting the perch or that we could measure? And this is a study back from 2013, 2015, right about when I was coming to the conference in the first place. And we were looking at all these different chemicals coming out. We watched them going through the wastewater treatment facility and then out into Lake Michigan. And we looked at measurements going through the dominant water patterns, so to the east and to the south. But of course, depending on water movement, they could go in various places. And some of the things were things that were standard that a lot of folks were measuring. Caffeine was the number one thing people were measuring at that time and using it as a wastewater treatment plant tracker, basically, because so much caffeine is used in the population and comes out of the wastewater treatment plant. It's processed, to a good deal by the wastewater treatment plant, but because we're using so much, a little bit gets out, and a little bit in this case is quite a bit. But the number one thing that we found that was really wacky was this. So each of these boxes is 25 nanograms per liter, and one of the things that we found is this compound, and it wasn't really on our radar, but they had just started measuring at our state lab of hygiene, was metformin, which is type 2 diabetes medication. And so, of course, I locked in on that. I'm like, what is this thing? And started looking at what are the metabolic impacts of this on perch, because it's a type 2 diabetes medication. You think it impacts metabolism. And so we looked and we weren't really finding anything changing in perch with the metabolic state. We were looking at calories, taken up and used, and then gene expression associated with different metabolic pathways. But a couple years later, we started reading about this medication being used for polycystic ovarian syndrome, which is a metabolic disorder that leads to reproductive changes. And so we started studying the impacts of that in the perch. And it just goes to show that these medications end up pushing all sorts of different pathways. And you just really, we're not really adequately describing probably what these chemicals are doing. And here's just an example. So here are some of the other things that were coming out. We have antibiotics. We have anti-diabetes medication. We have antipsychotic medications, anti-seizure medications, all sorts of things coming out of the wastewater treatment plant. But it's really, if you talk about how much is coming out, it's really below the therapeutic dose if you're looking at a per liter basis. But if you look at the discharge in pounds per year, it's actually quite significant. And if these things really don't break down rapidly, then this stuff could persist in the environment. And at that time, there were really only partial information about the effects that might be there. So my student, Ben Blair, went and did a risk quotient, as the EPA does, of each of the different chemicals. We measured 56 chemicals, but you can see there's not 56 chemicals up here. And part of that is either they really didn't cause any significant impacts, or we just didn't have enough data in order to make any kind of risk quotient assessment. But quite a few of these things, when you look at multiple sites, ended up going above 1, the risk quotient that we care about, and 0.1 where it's of concern. And so even just based on the data we had, there were several of these that might, at several sites, that might be of concern. But again, it only gives us part of the story when you don't really have a lot of information about all the different impacts these things might cause. One of the other things that has come out more recently, so in 2019, there was a study by Rose et al., and what they were trying to do is take those chemicals that they were finding in different water samples, and then using that big database that I was talking about from the Environmental Protection Agency, and trying to make predictions of collectively, if you're looking at all of those chemicals and what we know from high throughput screening of the potential molecular impacts these things might have, where would you see an impact in different watersheds? And I think this is moving towards a more predictive way of using molecular data in an environmental context, and then going and looking and seeing in those watersheds, are you really seeing those kinds of impacts? Which I think is a really fascinating way to look at it. One of the things that we've been working on is, can you look at particular pathways that are impacted in a particular stream or a particular waterway, and use this information to really collectively look at the suite of chemicals and look at pathway impacts. One of the ways we do this is through next generation sequencing, and as well as metabolomics. And I'll just talk about the next gen sequencing here. But for a while now, we've been looking at what pathways in organisms are being changed with single or multiple chemicals or with environmental samples. The benefits of this: you have an unbiased measurement of processes in cells, and no predetermined pathways that you're measuring, so you can look at basically collectively the global response. But the drawbacks are you're really providing a snapshot in time. Whenever you took that sample in the organism is what you're looking at as far as metabolism goes. It's not necessarily tied to a change in the phenotype of the organism. So you could see lots of changes in genes, but it doesn't really tell you whether the organism has been impacted. It's also a large quantity of noisy data. So you really have to be careful about the conclusions that you're drawing from genomic data. But we're trying to get around this by tying it to phenotypic endpoints. And linking this chemistry, linking the data that we're getting to the biological effects. My student Emma, and this was the collaboration that we ended up doing with Dana Kolpin that I had talked to at that conference, out in Iowa in Muddy Creek, which is a wastewater dominated stream in Iowa. So we collaborated with Dana and with researchers at the University of Iowa to look at the chemistry that was coming out in this particular stream, looking at any kind of biological impacts we were seeing in an organism, and then tying that to the gene expression data that we got from organisms in the stream. Particularly, we basically put cages of fathead minnows in the stream, let them sit there for 2 to 4 weeks, and then collected data from them to look and see if we saw any particular pathways that were related to some of the phenotypic effects in the data. And what we found is some of the candidates that we see all over the place when you're looking at wastewater treatment effluent. Metformin, again, was very high in the stream. We had venlafaxine, which is, again, a psychotropic medication or a cytological medication. Guanylurea, which is a byproduct of metformin in the wastewater treatment plant process. The bacteria convert it to guanylurea. And then fexofenadine is another one that's brain active. And we started seeing particular pathways as a result of this. First of all, you could see differences in upstream, which is this US1, and the effluent right outside of the effluent stream, and then downstream from the organisms in the stream, or the effluent in the stream, and both downregulated and upregulated. And we started looking at not individual genes, but groups of genes that were related to particular function. And we did see changes in the gene pathways that were associated with things like muscle contraction and signaling in cardiomyocytes. But we also saw things that were related to some of the psychological medications. So neurological pathways being stimulated in the brains of these fish. And this was with another one of my students, Peter Schumann, who's moved on to the Environmental Protection Agency and now over to Europe for a PhD, further looking at the implications of gene expression related to phenotypic changes that we see. So we can use this data in order to really show that there was some kind of impact on these fish as a result of the exposure to these chemicals. And it's a sensitive measurement in that you don't necessarily, we're not necessarily measuring for these things in our traditional toxicity tests, but it can give us an idea of some of the sublethal impacts of these chemicals, even in a natural environment, especially when we tie it to the chemicals that we're seeing that we know have that kind of an impact. One of the things that Peter did is look to see, used co-expression network analysis to take not just single genes, but group them into modules that looked like, or that were related to particular functions within the organism. So this is an even more, a better way of aggregating this information. So you're not just looking at individual pathways even, but using hierarchical clustering in order to analyze and really identify phenotypic things that could be changing in the organism. Sorry, it's taking a minute. And so looking at the modules that developed, some of the things that he found, and he was looking at the brain tissue in particular, were some of those neurological pathways that were disrupted. So it was related to things that we see in humans when we see changes due to Parkinson's disease, Huntington's disease, Alzheimer's, and neurodegenerative pathways. And so, which is really interesting. It's not to say that those fish are getting Parkinson's disease, but some of the dysfunction is related to neurological changes that could be related to those chemicals. One of the other areas in my research has been exploring new chemicals that, so emerging contaminants can be of kind of two groups. One is groups of chemicals that have been out there for a while and we just don't really know the impacts, or we haven't been able to measure them in the environment until recently and we realized they were a problem. And then we have new chemicals that are coming on the marketplace all the time. So manufactured nanomaterials, again, right when I was starting my job as a faculty member, started becoming important in developing new technologies. And I became a PI in a Center for Sustainable Nanotechnology, where we were looking at the impacts of various manufactured nanomaterials that were up and coming in the marketplace and how their chemistry ends up influencing how they interact with organisms. So I was working with chemists, including Kathy Murphy at the University of Illinois, and looking at these nanomaterials that the chemists were making and could tune to have different properties and how that might impact in my case, aquatic organisms. Also looking at the way that the chemistry of the surface ends up changing how it interacts with the membranes within various organisms, including bacteria, Daphnia, and Drosophila. One of the things that we saw when we were looking at these nanomaterials is that there were differences in sensitivity of organisms to these nanomaterials going across different species. Now, when we look at, we do this in toxicology, right? We look and see, we base our regulations on the most sensitive organism, but often we're not asking the question as to why is that organism sensitive and other ones are not. And so this is, again, where genomics can play a role and really tell us how is that chemical interacting with the organism and what makes that organism have that kind of reaction to that particular chemical. Often we find that Daphnia, over to the left of this diagram, are the most sensitive, along with some fish species, not all, and that other organisms, ones that are used to pollutants like chironomids, which are midges that are kind of a base of a sediment food chain in the aquatic environment, also are the least sensitive to a lot of toxicants. They're living in an anoxic environment and really are not so sensitive. And what we did is we used genomic data to look at a cross-species analysis and look at the most sensitive Daphnia all the way to chironomids, which are not so sensitive, and see which pathways were being implicated in those different species. We knew from our toxicology experiments also that there were certain pathways that were being pushed that we could see in the organisms. For instance, some of the heme-related proteins were changing the color of the chironomids. So chironomids are normally found as red in the sediments. They're trying to get they're dealing with the low oxygen environment with these heme proteins. But what's great is that that heme protein is red, and you can see it change color when it was exposed to things like metal oxide nanomaterials. And we found this was a whole pathway that was disrupted because of the nanomaterials. And so what we did is we measured then across the different organisms, could we see those heme proteins? And in the more sensitive ones, were they more affected? And that's exactly what we found. So the heme-related proteins in the Daphnia were changed negatively compared to the controls to a greater extent than the chironomids were. And the chironomids often had an opposite reaction to the nanomaterials and having more upregulated proteins to deal with them. And so it's a really interesting way of looking at providing lines of evidence for mechanisms for asking more questions about how these emerging contaminants are interacting with organisms. There has been this big move in toxicology towards in vitro data. So we are trying to move away from these whole organism assays and really look at not only normal contaminant or legacy contaminants, but emerging contaminants using cell-based assays. The problem is that the in vitro tests don't necessarily give you the same data as a whole organism assay. So it might be a place to start or give certain information, but it's not equal to the in vivo responses that have all these different organ systems that are interacting with each other. It doesn't really tell you about the uptake or distribution in tissues. Molecular changes can be adaptive in those cells in particular, and it's not necessarily linked to survival, development, or growth, which we care about in the environmental organisms as the endpoints. But it might be a good screening tool. And so one of the things that we've done is move towards high-throughput phenotypic profiling. And the idea behind this, it's actually very heavily used in pharmaceutical research, and so really great for emerging contaminants research as well then, is using high-content imaging of cells to look at hundreds of snapshots of those cells at high resolution. So you can look down to the organelle level and see how the phenotype or the morphology is changing of different aspects of that cell. And as I said, it's being used in drug discovery. And now US EPA has started trying to use this for looking at other cell types, but they're usually using human-derived cell lines for this purpose. And so, and again, the cells in cell culture from human studies don't necessarily always give you the full picture of what is an organism doing. However, if you have an organism that is one single cell, such as some of the algae, it's a great model organism for using this type of technology. And so there's a gap in knowledge of how not only the LCO, which is a nanomaterial, impacts these algal cells, other than the traditional measurements that we take are for toxicology are on living or dead, or are they affecting photosynthesis? And so we started using this high throughput phenotypic profiling for algal cells. And we looked at nanomaterials to start out with. We know that, again, we looked at this lithium cobalt oxide, one of the nanomaterials, and found that it decreased carbon dioxide assimilation in this algae, which is important as an ecosystem function. But the question is, what exactly is the chemical doing in the algae? And what we did is we use this cell profiling assay is using different stains to look at the different parts of the cell. So you can look at chloroplasts alone just because they fluoresce, but things like lipid droplets in the cell and the nucleus, you add a dye and it allows you to see the structures of those with these microscopes. And so then what you do is basically kind of facial recognition profiling, but of cells, and it tells you how the cell might be changing when it becomes exposed to a particular chemical. And so we started this kind of not knowing whether this was going to work or not, but the fact that it's being used in the pharmaceutical industry we thought was impressive. We developed this MACI assay as a proof of concept study and so we saw that cells were changing, but we weren't really sure if that meant anything. Again, like the genomic data, you don't know if it means anything unless you could tie it to something else. And so what we did is we picked chemicals that we knew the mechanism of action and compared it to chemicals where we didn't know the mechanism of action on algae. And then what we could see were phenotypic profiling changes that related to the mechanism of that particular chemical first. But then also we could see that particular chemicals that we didn't know also had similar kinds of profiles. And we did further study biochemically. We were able to see that that was really true, that chemical was acting in that way. So for example, the nanomaterials using this particular way of looking at this phenotypic profiling, we found that there's an indication that all nanoparticles are acting by oxidative stress. And so that was our first hypothesis, that it was going to look like the hydrogen peroxide here. But what we ended up finding is that we found in that previous study phenotypically that it was affecting photosynthesis. And what we found is that it actually looks like the photosynthesis inhibitor. And so the nanoparticle phenotype of the endpoint that we were interested in, the photosynthesis, actually was what was affected when we looked at this phenotypic profiling. So it's kind of a proof of concept. And we've been doing this now with all sorts of other chemicals. Here's just the example of the different herbicides, for example, that we were using for this. But we are now using this with other compounds, such as plastics and plasticizers and PFAS and other chemicals, to look and see how it overlays on this phenotypic profiling based on mechanisms of action that we already know. One of the ways that we're doing using this particular method is looking at nanoplastics. We're trying to, through a separate grant, look and see how nanoplastics relate to what we know already about some of the manufactured nanoparticles after studying them for a couple of decades. We know that plastic pollution is persistent. I won't go into this too much because you're going to hear all sorts of talks about plastic pollution, but we know that this is a problem in the environment. My student, Lizette, who is just graduating right now, was looking at different plastics that had different surface chemistries. Again, looking at data similar to the way we were looking at the nanomaterials before, where how the chemistry impacts how the nanoparticle influences the organism. And so she started out with polystyrene, which isn't probably the best model, isn't the best for looking at environmental impacts. It's not the most prevalent. However, we were able to modify the surface chemistry. And I won't mention all the data she has, but she's looked at other particular plastics as well. We know that these things can cross biological barriers, and so they're of particular interest in these studies. And we know that the chemical composition of a plastic could make a difference, not just the core of the nanoparticle, but what is on the surface. So again, changing those charges could make a difference. but also the polymer additives. So there are all sorts of polymer chemicals that end up in these plastics, and we only know about a couple of them and how they might impact organisms. And so was using rainbow trout gill epithelial cells. The first thing that comes in contact with chemicals in the environment and the fish is the gill cells. And so using that cell culture as a mechanism to look at toxicity and using these model nanoparticles, both positively and negatively charged, and then the plasticizers that go along with them to look and see what kind of impacts they have. One of the things that she found is that there were the positively charged nanoparticles, which just like before had the most impact on cell viability after 24 hours. But then she went and looked at differences in the morphology of the cells to see what other kind of impacts we were seeing. And she found differences in the nanoparticle treatments after 24 hours. These are some PLSDA plots showing differences. The positively charged particles had the greatest difference from all the other ones. But one of the other things that it pointed out was that the mitochondria were the target of these nanoplastics. And so she started really focusing on mitochondrial morphology and also the function. So first we saw changes in morphology in mitochondria, and then she went and looked to see, are we finding them there? And she found co-localization of the plastics in the cell close to the mitochondria. And then what she looked to see is, again, are we seeing the phenotypic change beyond just the morphology change in the cells? And she found that respiration was inhibited in mitochondrial bioenergetics. So this really is a target of these particular nanoplastics. She then also went through and looked at some of the plasticizers, and that work is in progress right now. But she's looking at not only the traditional plasticizers like DEHP, but some of the alternative plasticizers that people are now using because they're trying to replace one that we know is toxic with something, the other ones that we don't necessarily know the toxicity of. But in general, there's limited research on these emerging alternative plasticizers, so the next emerging contaminant. And we do see some similarities and differences going across. This is just preliminary data of hers. But here you can see the different dyes. So we have the nucleus, the endoplasmic reticulum, actin, and mitochondria. And the plasticizers have different clustering depending on what they are, and the concentrations. And so we do see plasticizers clustering together, and we're starting to decipher what these mechanisms of action are. But one of the things that she noticed is that actin seems to be impacted in these plasticizers, especially the ones that seem to be more toxic. Now, the cells seem to be growing different structures inside, and it's really interesting to see these changes happening and not really exactly knowing why they're happening. But we see this disruption to the cytoskeleton and the endoplasmic reticulum with particular plasticizers, where some of them have a great effect, these ones over to the right, and then other ones don't really seem to have any impact at all. So those really might truly be the least toxic alternatives. And again, these group based on largely their chemistry. So we can see that differences in the structure of the plasticizers lead to particular groupings of these plasticizers, and we can do these dendrograms, really clustering these things based on function that they disrupt within the cells, which is really great. It will lead to a lot more information about data on these plastics and plasticizers that will help us protect the environment. So these new tools in our toolbox hold promise to address these complexities. So we can identify the most likely candidate for impacts in a mixture of exposures, identify environments that need greater monitoring or cleanup, even using these molecular tools, identify which species are most sensitive and why, and create a better suite of high throughput tools for prediction for environmental safety. And so I'm looking forward to seeing how the scientific community uses this toolbox going forward. I think we've already made leaps and strides in the 10 years since the last time I gave a talk at this conference. And I look forward to seeing how other folks are using these tools. With that, this is just a smattering of the funding that I've received. Not everything is on here, and not all the students that I've worked with are on here, but these are some of the ones that are highlighted in this talk that provided data for the talk. And with that, I'll thank you, and I'll be happy to answer any questions.
Thank you, Rebecca. So we have two microphones in the room. Hannah and Lee each have a microphone, so if you have questions, you can raise your hand or you can hop up and they'll bring you a microphone so that you can ask it. And if we have any virtual attendees that have questions, feel free to type them in, and we have someone monitoring the chat, and we'll make sure to get those asked. So feel free. Any questions?
Yeah, thanks for the presentation. Going back to the perch, did you at all evaluate increase in the population of predatory fish in Lake Michigan when evaluating the perch decline?
Yeah, really good question. And working with the DNR, I was really focused in on the metabolism of the fish, but I know that they have done that and looking at what's going on. One of the fish is maybe not large predators, but I know that the round goby, which is an invasive species that came from Eurasia, has been eating eggs of all sorts of different fish in the Great Lakes, and the perch being one that has been shown by a couple of colleagues in my school, John Jansen and some other folks, that they're definitely a target. So there's definitely some impacts of not only the zebra mussels, but other invasive species coming in for eating the perch. Yeah, so I would never say that chemicals are the whole cause, but they were asking me, especially because of that weird gonadal structure, like what's going on there? It was more the question.
Other questions? Actually, just to follow up then, so how do you tease those apart? Or can you tease those apart?
Yeah, so what we tried doing, this is where maybe some of the molecular tools fall down. Without an annotated genome, it's hard to figure out so let's say we have a rat and a mouse genome. You try to translate what the genes are in that organism to perch, let's say. And so we're getting closer and closer as more people sequence and annotate different species other than the human models. But we still fall down in some cases as to what those genes are that are being turned on or off in a particular organism and how to use those to tell us what's going on. With the perch, what we ended up doing, we tried going out and sampling males after they showed us that result, and the male population was decimated. Like there was nothing. There was only females out there. So it was really hard to tell what was going on. We did a pilot study in our lab, which didn't go further because we started switching to fathead minnows, which are a little easier to deal with. But in the perch, we did an exposure of perch to metformin. They were adult perch, and looked to see if any of the gene expression markers or protein markers changed that related to endocrine function. And what we did see was vitellogenin showed up in males, which is a sign of endocrine disruption, and it's not supposed to show. Vitellogenin is, there are probably other folks in the audience that measure this, but vitellogenin is a marker for females. That's a glycolipoprotein that's deposited in eggs. So males should not be producing that really in any amount. And we were finding a significant increase in vitellogenin in those males. When we switched to the fathead minnow, and I didn't show this data, I showed it 10 years ago, but not this time. What we find in fathead minnows, which is a much smaller fish and a little easier to deal with as a model than a perch, when we expose them as adults, we see that same response. The vitellogenin in males shows up. And then when we do exposures going from babies all the way through adults, we see changes in the structure of the gonads when exposed to metformin, to the point where we saw ova being produced in the gonads. So it really could be something that indicates that that might be what's going on with the perch. It might not explain the whole thing. There are a whole suite of chemicals that are coming out of that sewage treatment plant, but it's definitely one that's in abundance, or was maybe up until I haven't done a recent measurement to see how metformin might be decreasing with these new GLP-1 medications that people are using. I'm really curious as to how those GLP-1s will affect metabolism in fish. But definitely the metformin was dominant as far as emerging contaminants coming out of our wastewater treatment plant up until, probably 5 years ago.
That's really interesting. Yep, we have one more question here.
Hi, I'm a person who takes metformin and acetaminophen, of course. And is there any way that your research can tie into the wastewater treatment plants to reduce the concentrations of those pharmaceuticals going into their waste system?
Yeah, that's a great question. We have collaborated with Veolia, which runs our wastewater treatment plant here on behalf of the Milwaukee Metropolitan Sewage District. And we were looking at things, actually nanoparticles that you could add as a final treatment step that would filter out these medications. The sewage treatment plant does a good job of getting rid of a bunch, dropping the levels of a lot of the medications, but there are some ones that are really just not so great, and we're using them in such a quantity that they still are coming out. And so metformin is one of those where it's kind of partially taken care of, but it's such huge quantity it comes out. So we were looking at nanoparticles, kind of like a water filter, like you use in a Brita filter, excuse me, in your kitchen, but in a really tiny form. So it was like grams worth of these nanoparticles, filtering out the final stages of the effluent before it went out into the lake. Did a really good job of absorbing those chemicals. And of course, we also have nanofiltration, but it's really expensive to put on a wastewater treatment plant. They are doing that in Paris, for example. I went over and talked to their water company, and they have that nanofiltration as part of their treatment process because they have such a short line going from effluent to drinking water uptake, that they need to take care of it somehow. So these huge operations to do that. But one of the other things is to maybe really evaluate those pharmaceuticals before we start using them. Is that really the best one to use? And is that going to cause any kind of environmental impact? If it's pushing a particular pathway in a human, is it going to do the same thing to a negative consequence in a fish or in another organism? And so I've been talking to policymakers about trying to figure out how to educate doctors, and then also how to really start research programs related to that potential impact. So I would never want someone to take a medication that's really helping them. Obviously, metformin helps a ton of folks. One of the problems with metformin, though, is that it's prescribed just like the GLP-1s now for everything, like all the way up to from the diabetes to it started being prescribed for estrogen responsive breast cancer. And I've spoken to several breast cancer researchers. They're like, no, it does not work for that. And people are prescribing it for that. And then also for polycystic ovarian syndrome, again, it helps women that have that insulin resistance. But then also for anti-aging, it increases antioxidants in the cells. And so it's being prescribed for a population for anti-aging drug. So this huge swath, even if you just took the diabetes, folks with diabetes, it's being prescribed for pre-diabetes, and it hasn't necessarily been shown to prevent diabetes from occurring by taking that medication. And so someone estimated that the state of California, it was something like 70% of the population could be really legally prescribed this medication just based on the pre-diabetic condition alone, which is crazy. Can you imagine the whole population taking one medication, no matter what? That's a huge amount of that medication going out. And like I said, I have no idea about the GLP-1s now, what the implications are for that, since so many folks are taking that. So, but again, I wouldn't want to stop folks from taking a particular medication, but I think we really have to enter other conversations about environmental impacts of our health choices. That was a long answer. Sorry. That's one. I didn't talk about that so much here. It's in my other talks that I gave previously. So, yeah.
Dr. Klaper, I'd like to follow up on that actually, because I very much agree with you with what you've proposed here, but what that is, and even maybe expanding that to all contaminants approved for industrial application, that is a massive paradigm shift from how it is currently approached. So how, in your opinion, can we walk this path? How can we push this forward as environmental scientists, engineers, to make change in a timeline that's actually going to make sense for the sustainability of humanity and our Earth?
Yeah. Oh, it's a really good question. And yeah, I struggle with that. It's like, what are the baby steps to get us there? I don't know. I think that starting to have that conversation out loud is really important, and having the scientific community start pushing that, and policy community start pushing that idea. I would say the REACH, regulations in Europe are a really interesting way that might get around some of it, or may address some of it, in the fact that they're regulating based on volume of chemical being produced and used. And instead of, with us, some of the chemicals go through Food and Drug Administration, some go through EPA, and sometimes those things don't touch each other. Other things that aren't used for consumption aren't necessarily evaluated in the same way. We have this hodgepodge of chemical regulation here, which is kind of no, it's not serving a good purpose, I don't think. So that might be another thing, is trying to push to have a different way of looking at chemicals, which again, it'll be hard here, but I think it's necessary. But another way is, again, I think baby step is really talking to the medical community for the pharmaceutical end of, that whatever they're prescribing is having an impact in the environment. And we're not necessarily having that conversation. The medical students aren't hearing that message. And so I think that could be a small place to start. I've started talking to legislators here about having that be part of a medical system. We have several legislators in the state of Wisconsin that are nurses or doctors. And so I've been trying to talk to them about maybe having that be part of an education system for the medical community. And that might be a place to start. Small step.
Yeah. Other questions? Okay. Going once, going twice. Okay, thank you, Dr. Klaper. That was really great.
Yeah, great questions. And thank you so much again for having me here.